Welcome to session 34. The topic of today's discussion is mechanism of enzyme action. So guys, in the previous lectures, yeah, uh, so uh, we need to discuss today the mechanism of enzyme action, how enzymes are going to act on the substrate that we need to see. Uh, all chemical reactions in a biological system have an energy barrier which prevents reaction from proceeding in an uncontrolled and spontaneous manner. The input of energy required to break this energy barrier or to start a reaction is called as the energy of activation. So it is designated as EA. Enzymes lower the activation or energy of a reaction, that is required amount of energy needed for a reaction to occur. They do this by binding to a substrate and holding it in a way that allows the reaction to happen more efficiently. So this is an illustration of a enzyme substrate complex that drives specificity. So usually, as I said you, the enzyme is a very large molecule. It is a large molecule, and the substrate is relatively small. So this is the substrate, and so this particular area is called as the pocket, or we call it as an active site, right? It is called as an active site. Yeah, this is a reaction. So this is a diagram that shows us where we can explain how a substrate gets converted to product, and we can see there is a transition in the energy. So the substrate, the free energy increases of the substrate. So this we call it as an energy of activation. The substrate has to increase its energy of activation, and then it forms an intermediate complex, and then it gets converted into it gets converted into a product, right? So so the substrate initially does not, it is not directly getting converted into product. The substrate increases its energy of activation, and it reaches to a particular state called as transition state. Then the energy of activation decreases as soon as we get the product. Yeah, so this reaction is in the absence of an enzyme, the energy of activation is more. So this is more, and in presence of the enzyme, the energy of activation is less. So here we can see a reaction that is shown, which is shown in blue. It is in presence of enzyme. The substrate increases its energy of activation, which is a very small peak we can see here, and the substrate gets converted into enzyme substrate complex, and then we get enzyme and product, and then this is how the peaks are very small. In case of the delta G is very small for the transition state. In absence of the enzyme, the energy of activation to reach the transition state is much higher. Okay. So activation energy, it is important to remember that enzymes do not change the reactions delta G. In other words, they do not change whether a reaction is exergonic or endergonic. This is because they do not change the reactants or products free energy. They only reduce the activation energy required to reach the transition state. That is what we have seen in the diagram. Enzymes bind transition states best. The idea was proposed by Linus Pauling in 1946. Enzyme active sites are complementary to the transition state of the reaction. Enzymes bind transition states better than substrates. Stronger or additional interactions with the transition state as compared with the ground state lower the activation barrier. Yeah, this is an illustration of TS stabilization idea. So this is how this is the substrate. Imagine as a stick model. Uh, so in the transition state, we can see this stick is undergoing a bent. It is undergoing a bent, and the product is we get a broken stick. We get a broken stick. So, so the energy, the transition state, the energy required for the to reach the transition state is quite high. Yeah, in case of in the presence of an enzyme, so enzyme is complementary to the substrate. So our substrate, which is like a stick model, this substrate is bound by an enzyme, like the magnets, and then the substrate is getting converted into few products, and the uh, energy of activation is quite less. So enzyme complementary to the transition state. So this is enzyme substrate complex, and then we get enzyme, and we get the product. We get the enzyme and the product, and this is a comparative diagram that shows the difference in the transition energies. So in absence of the enzyme, the transition energy is quite high, and in the presence of the enzyme, the transition energy uh, is quite low. Enzymes may use one or a combination of the following catalytic mechanism. It can use acid-base catalysis, so which is give and take protons to break down the complex bonds, or it can use covalent catalysis, so where there is a change in the reaction paths, or metal ion catalysis, which uses redox cofactors or the pKa shifters. So these are my references. Thank you one and all.